Welcome to The Explainer. Today, we're tracing a really high stakes, literal life saving journey. We so often think of blood donation as just this simple logistical exchange. You roll up your sleeve, give a pint, and someone else receives it. Easy, right? Well, on a microscopic level, a blood transfusion is actually an incredibly profound biological handover. We are literally transplanting living tissue from one human being to another. And to make absolutely sure that journey is safe, modern medicine relies on an incredibly rigorous chain of events. So, get ready because we're going to see exactly what happens when your body encounters someone else's blood.
48. Think about that number for a second. When we talk about blood, we usually just picture a simple red liquid. But scientists have currently identified a staggering 48 different blood group systems in humans, making up over 350 different antigens. That's not just a red liquid. That is an enormous amount of cellular diversity hiding right there in our veins.
Okay, let's dive into this, because this right here is the key to our whole explainer today. If there are literally 48 distinct systems mapping the surface of our red blood cells, why on earth do we only ever hear about the ABO system and whether we're positive or negative in the RH system? Today, we're unpacking the medical protocols that answer that exact mystery. So, here is our roadmap for today. 1. The transfusion chain. 2. Cellular identity. 3. The ABO system. 4. The rhesus system. And 5. Safety and compatibility. We're going to follow this path to completely demystify this complex biology.
Starting with Section 1, the transfusion chain, the physical journey from donor to receiver. Before we get into the microscopic stuff, we have to look at the real-world logistics. This is the chaîne transfusionnelle, the 4 vital links connecting donor to receiver. First up, collection. That's the physical donation. Step 2 is qualification, which is the crucial biological testing phase. Step 3 is processing, where the blood gets separated into specific products like red cells, plasma, or platelets. And finally, Step 4, the transfusion itself. Every single drop of donated blood must successfully pass through this exact sequence.
But hold on a second before we move down that chain. 4%. In places like France and similarly monitored populations across the globe, only about 4% of people donate blood on a regular basis. Think about that. That whole physical chain we just talked about, it relies entirely on this tiny, tiny fraction of volunteers. It makes every single collection incredibly precious, which is exactly why the science of ensuring absolutely none of it is wasted and all of it is perfectly safe is just absolutely crucial.
Section 2, cellular identity, the blood's immune system. Let's zoom in right now.
We're looking at that second step of the transfusion chain, the qualification step. How do our bodies actually distinguish our own blood from foreign blood? Well, you can think of antigens as cellular ID cards. They're these complex molecules, mostly proteins or sugars, sitting right on the surface of your red blood cells. Now, think of antibodies as your immune system's border patrol, floating around in your blood plasma. Under normal conditions, your body is totally cool with its own antigens. But if foreign ID cards enter the bloodstream and they don't match what the border patrol expects, those antibody security guards are going to attack and destroy the invaders immediately.
Historically, not knowing about these cellular IDs made blood transfusions incredibly dangerous. It was basically a fatal roll of the dice. It actually wasn't until 1900 that Carl Landsteiner discovered the ABO system. And it took almost another 4 decades until 1939 and 1940 for Levine, Landsteiner, and Wiener to finally map the rhesus system. When you zoom out and look at the vast history of medicine, these absolute game-changing, life-saving discoveries are astonishingly recent.
Let's move to Section 3, the ABO system, the universal IDs.
And this brilliantly illustrates exactly how your baseline cellular identity works. Your blood group perfectly defines your biological defenses. If you're Group A, you've got Antigen A ID cards. So, naturally, your plasma carries Anti-B border patrol. If you're Group B, it's the exact opposite. Now, if you're Group AB, you have both ID cards, which means you have absolutely no natural border patrol, making you a universal receiver for red cells. And if you're Group O, your cells have 0 ID cards, meaning your plasma has to carry both anti-A and anti-B antibodies. The crazy thing? These are natural antibodies, meaning they develop shortly after birth, even if you've never been exposed to foreign blood.
So what does this all mean? It brings us to the golden rule. You must never, ever transfuse red blood cells bearing antigens that the receiver's antibodies are programmed to attack. If a Group O patient, whose blood is
literally loaded with anti-A and anti-B patrol units, receives Group A blood, the consequences are immediate and severe. It causes massive, life-threatening red blood cell destruction, which is medically known as hemolysis. You just cannot bend the ABO rules, period.
Section 4. The Rhesus System, the hidden danger.
Once we have ABO compatibility locked down, we have to look for a secondary, hidden threat. This was initially discovered through studies on Macacus rhesus monkeys, hence the name. The rhesus system is super complex, with dozens of its own antigens, but the most immunogenic, the really powerful one, is antigen D. About 85% of the population has this antigen D on their red blood cells. That makes them Rh positive. The other 15%, they completely lack it, making them Rh negative.
Now, what's really interesting about this slide is the stark contrast in how our immune system actually handles ABO versus rhesus. Remember how ABO antibodies are those natural, regular IgM molecules you just have since birth. Rhesus antibodies are a completely different ballgame. They are irregular IgG molecules. That means an Rh-negative person doesn't naturally just walk around with Rh antibodies. They only develop them if they are accidentally exposed to Rh-positive blood. They act much more like specialized military units that are only drafted after an initial foreign invasion. And this reactive nature creates incredibly high stakes, especially during pregnancy, through a mechanism called fetal-maternal alloimmunization. Picture this. If an Rh-negative mother has an Rh-positive baby, their blood might mix a bit during delivery. The mother's body recognizes that foreign D antigen and goes, uh-oh, threat, and creates those Rh antibodies. During a second pregnancy with another Rh-positive baby, those smaller IgG antibodies can now actually cross the placental barrier and attack the fetal red blood cells. But here's the good news. By understanding this exact biological mechanism, modern medicine can now prevent this entirely. We just inject the mother with specific immunoglobulins to stop her body from ever forming those antibodies in the first place. Incredible, right?
Section 5. Safety and compatibility. Securing the chain.
Let's zoom back out to that overarching framework we started with. Securing the blood supply isn't just about matching up ID cards and border patrols. During the biological qualification step, every single collected pouch of blood undergoes an absolute, non-negotiable safety net of infectious disease testing. We are talking screenings for HIV, hepatitis B, hepatitis C, syphilis, and we even measure liver enzymes like ALAT to ensure total patient security. If any single test flags positive, that unit is immediately destroyed. No exceptions.
But beyond infectious diseases, we also kind of have to protect the receiver from themselves. Remember those 48 blood systems we mentioned at the very beginning? Yeah, we can't perfectly match all 350 antigens every time. So before a transfusion, we do what's called the RAI test on the receiver. We are actively checking their plasma to see if they've developed any sneaky, irregular antibodies from previous transfusions or past pregnancies. If we spot an irregular antibody against even a minor blood group, we have to go find a specific donor pouch that totally lacks that minor antigen.
And finally, even after all the lab testing, the blood typing, the infectious disease screenings, there is one last dramatic step: the ultimate bedside check. Literally right before the life-saving transfusion begins, the medical team performs a final cross-match, right there at the patient's side, physically verifying the donor blood against the receiver's identity one last time. Only when this final check passes does the biological handover begin. Which really brings us to this profound Arabic proverb: a drop of blood is better than a treasure of gold. When you really understand the sheer biological complexity behind it, the 48 systems, the antigens, the antibodies, the unbelievably precise logistical chain required just to move a single drop safely, it proves exactly how true that saying is. Your cellular identity is entirely unique to you, yet it holds this incredible, almost magical power to seamlessly sustain someone else's life.